Okay, guys, ready? Doesn't matter if you're not. We're going anyway. Chapter 5 is on obviously homeostasis and transport. Just a quick review on homeostasis. Let me get out my pencil. So remembering when I told you about vocab in biology, it's all about breaking down the parts of words because they have a translation. Oh my God, and every time I talk to you guys, I keep reminding myself of that stupid periodic table video that I have. Eventually,
Oh my God, it's been many times.
I know. I'm so bad. Okay, I will try to remember again, but you know what? I'm just not gonna promise anymore. It'll show up probably like just about as you're about to graduate. It'll be like, oh wait, here's the video. Anyway, breaking down parts of words because they come from Greek and Latin. So stasis is environment and homeo is stable. So homeostasis is a process, because that's the cis part, the process of maintaining a stable environment. And a great example of that is body temperature. Body temperature is always relatively constant unless you're sick and have a fever. So that requires a lot of energy in order to maintain that. Plus we have, for mammals, we have hair or fur. Birds have feathers. So there's a lot of adaptations that allow us to maintain homeostasis. There's a lot of other ways that all organisms maintain homeostasis, but body temperature is just the easiest one because we are all familiar with that concept. Okay, now, transport is used to maintain homeostasis. So for example, when your cells use energy, they produce waste products, and the waste products have to go out. So in your, I'll just go back to the, yeah. So in your cell, you need ATP to go in and you need waste product to come out, as well as you need oxygen to go in and you need CO2 to come out. So transport is how this is going to happen. This is how your body maintains homeostasis. You've got homeostasis between the gases, and you have homeostasis between energy and the waste products from what's called respiration, which you'll learn about later. But transport, like I said, is all about just being able to maintain this stable environment. There's going to be two main methods of transportation. And that's the chapter is broken down into two sections. The first section, which I'll talk about today, is passive transport. Passive transport does not require energy. That is the main characteristic. And the second type, which I'll talk about next week, is active transport. That does require energy. And hopefully the names are kind of, I don't know what the version of the adjective version, but the name implies that whether energy is involved or not. So passive is kind of like relax, whatever, whatever, and no energy needed. But active should tell you like, ooh, energy is involved. There's going to be 4 specific types of passive transport that I'll talk about today. They come in kind of, I like to do them in pairs because 2 of them are very similar and the other 2 are very similar to each other. I mean, so if you can understand how one of them works, you can understand how at least 2 of them work. 2 of them work. I told you my brain is like, I don't know, gone somewhere. All right. So keeping in mind that the cell membrane is the cellular structure that's going to control all of this, because the main function of the cell membrane, besides just being a physical barrier, is to control what things can go in and out of the cell. So the movement of substances, and I should have specified without energy, is called passive transport. So the first type of passive transport is called diffusion. I'm thinking you should probably have learned this with teacher Vanessa somewhere along the ways, not saying that you necessarily remember it or not, but it should be familiar. This is the simplest type of passive. Oops, that's a bad color to use. Oops, not writing simple again, passive. Simplest type of passive transport. And how diffusion works is it's the movement of molecules from a region of higher concentration to a region of lower concentration. Now, we've all seen diffusion happen. If you've ever seen a fire or somebody light up a cigarette or like any kind of smoke at all, you have seen diffusion. Because what happens if you're going to observe my beautiful art skills again. You see somebody who starts a fire. It looks like a coral reef, but anyway, that's my fire. And you start to see smoke coming off of the fire. So there's going to be a big concentration of smoke really close to the fire. And as time progresses, you're going to see that smoke start to spread out in all directions. If you're in a small room, diffusion is going to occur until the entire room is full of an equal amount of smoke. So this is diffusion. We've all seen it. If you've ever, like I said, if you've ever seen smoke, you've seen it diffuse. And I don't know if my GIF is going to work. Nope, doesn't work. Okay. Diffusion depends on there being a concentration gradient. Without a concentration gradient, no transport really occurs. Concentration gradient is when you have a space that contains a different concentration from one part to another. The easiest way to think of concentration gradient is like a hill where you've got a great concentration on one side and a low concentration on the other. And this makes it really easy to understand passive transport because it doesn't require energy. Just like if you're standing on top of this hill with a football, and you let it go, you don't need to actually exert any energy on the ball to make it go down the hill. It does it naturally. Okay, no energy required. The only thing that it needs is a slope or a gradient. And so this is the, I guess the, what do you call it? The, when you picture things in your head, the illustration, there you go. It's the illustration of the net movement of molecules in diffusion. It doesn't require any energy input at all. So as soon as the fire is started and smoke is produced, the molecules will naturally diffuse into the air.
Or out of the cell.
Let me just start that over.
Their net direction of flow into or out of the cell depends on the concentration gradient, the difference in the concentration of like molecules inside and outside of the cell. Oxygen will always diffuse into a cell because the oxygen concentration is always greater in the blood than in the tissue cells. The opposite is true with carbon dioxide. So carbon dioxide will always flow from cells to the blood.
Okay, so that is a good explanation of diffusion using gas exchange. So if this is inside your cell and this is the blood, actually I should do that opposite, right? Yeah, because it's red on the bottom. All right, this is the blood. This is inside the cell. So inside the cell, you have ATP being used up for cellular processes, and then the chemical reactions are going to produce carbon dioxide. So you have lots of carbon dioxide inside your cell. You don't have a lot of carbon dioxide in the blood, but you do have a lot of oxygen. There she is. You do have a lot of oxygen, and you don't have a lot of oxygen inside the cell. So you have a natural gradient between the inside of your cell and the blood. So naturally, as the blood flows by the cell, oxygen is going to diffuse in and carbon dioxide is going to diffuse out just because of the difference in concentrations. Now, when it gets to your lungs, the lungs are a different situation. Okay, you can stop. No. Nope. Next slide. Okay, when it gets to your lungs, let's see how I'm gonna do this. Here's the blood. And then this is, I'll use white. This is in your lungs. So now when the blood goes back to your lungs, inside the lungs is a lot of oxygen. Inside the blood is a lot of CO2. Cannot use green. And so the opposite process happens in the lungs. Oxygen is going to diffuse into the blood. CO2 is going to diffuse into the lungs, and you breathe that out. But it really is just any molecule that is concentrated on one side, which you can see here. It's going to go through a porous membrane, porous meaning that it has holes, and that's going to continue until you reach a point called equilibrium. Equilibrium is when there is an equal concentration across a space. The one thing that I do want to mention is that diffusion requires a moist membrane. Moist meaning like kind of wet. Okay, it doesn't happen in the body. I mean, like for cells. I mean, obviously, smoke can diffuse through the air without a moist membrane, but in the body, when you're talking about cellular diffusion, you have to have the plasma membrane there. Okay, so that continues until equilibrium. So high to low, like a ball rolling down a hill. Okay, remembering that the cell membrane is partially permeable, so only some substances can get through. The only substances that are able to diffuse through the cell membrane are going to have to be able to dissolve through the lipid bilayer. This means that they have to be nonpolar. So only nonpolar substances can diffuse through the cell membrane. If you have particles that are polar, they're going to have to go through proteins, those channel proteins that I talked about before, which I'll mention again when I talk about the third type of passive transport. But the really important thing to remember is that because the lipid bilayer is nonpolar and you've got your transport proteins here, the only substances that can go through the nonpolar lipids are the nonpolar substances. So two really easy ones to remember are carbon dioxide and oxygen, because they are also nonpolar molecules. So they don't have a problem diffusing through the lipids. Okay, the second type of passive transport is osmosis. This is, remember I said it comes in pairs. There's 4 of them all together, but 2 of them are very similar and the other 2 are very similar to each other. So osmosis is very similar to diffusion with one difference. Okay, water molecules. That's the only difference between osmosis and diffusion. Osmosis refers specifically to the movement of water molecules from high to low. And the reason why this is important is because your entire body is aqueous, right? The majority of the material inside your body is water. So it's very important to talk about how water moves from one place to another place. And of course, I found this funny comic. Why doesn't osmosis work like this when you've got your concentration gradient where you've got high info to low info and you just can't absorb it straight through the books? Nope, doesn't work that way. Okay, so diffusion is high to low with anything except water. Osmosis is high to low, only water.
Another type of diffusion is called osmosis. The left side of this beaker is filled with a salt solution, while the right side is pure water. A semi-permeable membrane separates the 2 compartments. It allows water molecules to pass through, but not salt molecules. In a solution, solutes, in this case the dissolved salt particles, take up space that water molecules would otherwise occupy. This results in a lower concentration of water molecules on the solute side. Because of this, Water will move by osmosis through the membrane from the compartment on the right with lower solute concentration to the one on the left with the higher concentration until equilibrium is reached. As a result, the volume of the right compartment decreases while the left compartment increases. So
Okay, so that's the effect of osmosis because the pores in this membrane aren't large enough for the salt molecules to diffuse to the right, but they are small enough for water molecules to osmose from the right to the left. So the height of the water on the left side increases. Now, the net direction of osmosis depends on the concentration of solutes on either side of that membrane. And there's going to be three types of solutions that will have a direct effect on cells depending on that concentration. So the first type of solution that I want to talk about is where the concentration of solutes is the same on both the inside and the outside of the cell. This is called isotonic, iso meaning same or similar. Oh, let's just go with same. So concentrations on the inside and outside are equal. Now, that doesn't mean that there is no movement of any molecules at all. Just like the diagram implies, it means that there is an equal amount of movement both in and out of the cell. So the important thing to understand here, there's a red blood cell up here. Being in an isotonic solution, it has no effect on a red blood cell because water will go in and out at equal rates. But if we put the red blood cell in what's called hypotonic, hypo meaning more than, that means that the external solution is hypotonic to the cell. That means that outside the cell, there is less solute, more water, because it's going to get confusing if we talk about concentration. And then inside is more solute, which means less water. So if you have more water to less water, water is going to move into the cell and you're going to have something called plasmolysis, which means that the red blood cell is going to explode. So we say for this particular example, we say that the external solution is hypotonic, or the outside, where should I put this? So outside the cell is more concentrated. And when I say more concentrated, I mean more solute, because there's different ways to describe solutions. So the concentration of solute outside is lower than inside, right? So less solute outside, more solute inside. If you have less solute outside, that means the concentration of water is higher. Because if you think about, let's say you have two beakers, this is 100% water. If I start adding salt or, sorry if you can hear that, that's my cat having a fit. If you add salt or sugar or anything to this beaker, it is no longer 100% water. It's going to be less than 100% because some of that volume is going to be taken up with the solute. So the more solute you have in the solution, the lower the water content is. So the net movement of water in a hypotonic solution is into the cell. Now there was a student from EP06 named Panat, and he came up with a really good trick to remember this. Hypo looks like hippo, and hippos are fat. And I think they have ears or something. And so if you just remember when the cell gets fat like a hippo, that's hypotonic. So I got to give him credit for that, because I told him, I don't know what year that was, like 2008 or whatever. I was like, I'm going to use that, and I still do. So if you guys ever come up with anything clever, let me know and I'll use it. And probably 10 years from now, I'll still be using it. Okay, so I said plasmolysis earlier. I confused my two cell states. This is cytolysis. Cytolysis is when cells burst. I think I've gone over wait, what am I doing? I think I've gone over phonetics before, but when you see this is in the book as well, I think. Let's see. Ow. Is it? Why is cytolysis not in there? Oh, interesting. All right, I guess not. I don't know why. I don't see it in your book, but anyway, you see it when you get to contractile vacuole, which I'll talk about in a second. Anyway, this is something called phonetics. It explains how to pronounce the word. I'm pretty sure I mentioned it before. And then the syllable that's in all caps is the one that's stressed. And again, with vocab, if we break down cyto and lysis, lysis is to break, and cyto means cell. So to break the cell. I thought I had a video, maybe a bit later on. Okay. And then the last type of solution is the opposite of hypotonic. That's called hyper. Oh, shoot. I am so sorry. Oh my gosh. I'm like so not. Oh, so stupid today. Hypo means less than. Okay, just to go back, less than. And that's why, because there's less solute, the external solution is hypotonic. I don't know why. Oh gosh, I'm so sorry. Okay, just to clear that up. Hypo, the prefix means less than. Now going to the last type of solution, that's called hypertonic. Hyper means more than. Okay. So if the external solution is hypertonic, that means outside is more solute, which means less water, and inside is less solute, which means more water, which means your net movement of water is out. Okay, so the cell shrinks. Okay, the concentration of a solute outside the cell is higher than inside. So the net movement of water is to the outside of the cell. Okay, so there's different ways for organisms or cells to deal with osmosis. I mentioned this in chapter 4 when I talked about vacuoles. This again is a paramecium. It lives in fresh water. So outside of the paramecium is fresh water, which means it has a higher concentration of water than inside the paramecium, which means that it's constantly absorbing water. Now, it's going to blow up just like a red blood cell will, unless it has a way of dealing with that. And it does. It has something called a contractile vacuole. So the contractile vacuole will fill up with water and why? Oh, there we go. We'll fill up with water and then it will contract like you just saw it do there to squeeze the water out. And as soon as it does, it will start to fill again, as you can see. And you can see here that there's more than one contractile vacuole. It's got one there, and there was another one over here somewhere. So for freshwater organisms, it's important that they are able to adapt to their environment. The paramecium, say like millions or even billions of years ago, that didn't have contractile vacuoles, they would have exploded and died. And because they explode and die, and like their DNA doesn't contain the information for contractile vacuole, that DNA will not be passed on. The only paramecium that will survive to pass on the DNA to the next generation are the ones that have the contractile vacuole. So then over time, your entire population of paramecium all have contractile vacuoles. Now, osmosis has a different effect on plant cells because of the cell wall. Plant cells do not explode because the cell wall contains cellulose, and cellulose is very strong. And I mentioned this before when I talked about the central vacuole and turgor pressure. when I mentioned like when you don't water a plant and the cell wall shrinks. And you can see here, this is a hypotonic environment. This is when you water a plant, and you can see the pink is the central vacuole. So the central vacuole fills up with water, squishes all of the organelles to the side of the cell wall, and you have what's called turgor pressure. This is what allows plants to grow hundreds of meters tall without a skeleton. Hypertonic is when you fail to water your plants. So the central vacuole shrinks, and you can see how much the cell membrane shrinks away from the cell wall. And this is when your plant wilts. That's when it basically is like bleh, like the leaves get all floppy. So this is going to be a time lapse of a wilted plant that's been watered, and now you can see the effect of turgor pressure over time. So the increased pressure from the full vacuoles is what allows these leaves to kind of perk up. And that last little one out there straightening out. Okay, this was plasmolysis, the one that I screwed up before. This is an example of plasmolysis. This is when the cell shrinks. Specifically for plant cells, when the cell wall shrinks away from the, when the cell membrane shrinks away from the cell wall, that's called plasmolysis. And that's when your plant wilts. I do have an experiment that I want to give you, and I'll describe it after I'm finished with this. Super easy, you just need potatoes. So these are plant cells under the microscope. And there's a plasmolysis. You see how they're shrinking? I really wish we could see this in the lab, but you know, COVID, blah, blah, blah. Otherwise, you could see this yourself. So this is plasmolysis. You see just how much the cell membranes shrink away from the cell wall when they're in a hypertonic environment. I do believe they're going to add more water so that you can see them expand in a hypotonic environment. Yeah, I can kind of, oh, buddy, slow down. Yeah, so that's the entire cell contents there, like within the cell membrane. There you go. Woof. Spread out again, hypotonic. Okay, so those are the first two. The next two are going to be similar to each other because they're going to involve a carrier protein. So the first one is called facilitated diffusion. Now, facilitate means to help make happen. That's an A. To help make happen. So recall what I said a few minutes ago about how the only molecules that can diffuse through the cell membrane directly are nonpolar molecules because they go through the nonpolar phospholipids. These, facilitated diffusion is used for molecules that cannot diffuse through the cell membrane. They are insoluble in lipids, or perhaps they are just too large to go through the lipid bilayer, but they are assisted by carrier proteins. These are going to be integral proteins, which I talked about. Oh, I can't write today. Proteins. Integral proteins, which I talked about in chapter 4. These are the proteins that go all the way through the cell membrane. They are completely embedded. So they have, they touch both the outside and inside environment of the cell. So what you're going to see in the animation is a molecule of glucose that is going to go through a carrier protein. When the molecule binds to the carrier protein, the protein will change shape and allow the molecule in or out of the cell. Now, the important thing about carrier proteins is that they are specific for the type of molecule they transport. What that means is that if this carrier protein is able to transport glucose from one side to the other, it is only meant for glucose. It will not transport any other molecule. Wait, you're supposed to play. Bruh. There you go. Okay. I was saying something. I was wrong. So it's not the blue one, it's the green one. I should have known from the color. Derp.
Other transport proteins act as carriers. When specific molecules attach to them, they change shape. The molecules are then carried across the lipid bilayer. Glucose molecules move across the cell membrane with the help of a carrier protein.
Okay, so he mentioned that the protein changes shape. It might be too grainy to see, but in the beginning, the protein is open to the outside. And you've got the glucose molecule, which comes in, and when it binds to the protein here, it closes to the outside and it opens to the inside, and that's what allows the glucose to go through. Many cells depend on glucose for their energy needs. These molecules are too big to diffuse easily across the cell membrane. So when the glucose inside the cell is lower than outside the cell, the carrier proteins will transport it in. Still important to remember, this is movement from high to low. So glucose is only transported into the cell when the level of glucose inside is lower than outside. And the last type of passive transport is called ion channels. Now, the reason why I said the last two are similar to each other is because ion channels are basically another name for carrier proteins, which participate in facilitated diffusion, but they are specific, obviously, for ions. So I suppose if you wanted to, you could say facilitated diffusion, and then a subset of that are ion channels, because it's still the process of facilitated diffusion, meaning the proteins help the ions diffuse through. And again, it is specific. So ions are really important minerals for your body, like sodium, potassium, iron, magnesium, zinc. All of these are ions that you get through the food that you eat, and they need to be transported into the cell. Now, because they have a charge, because remember, everything in your body is aqueous. When you eat salt, that breaks up into sodium ions and chloride ions. These two ions are super important for the functioning of your kidneys, and they are transported through your blood as ions. When they get to your cell membrane and they go to that lipid bilayer, your lipid bilayer is nonpolar. And I know I've mentioned before that nonpolar and polar don't mix, and polar is only being slightly charged. Imagine how nonpolar feels about an entire charge of plus or minus. It's like, it's really not getting through. So ions need carrier proteins to get transported from one side to the other. Again, sodium, potassium, calcium, I forgot calcium, and chloride. I actually took a class in university called biochemistry. The only thing I remember from that class is calcium is super important. It was like, there was so much chemistry in that. And all I really remember is calcium is like, goes from one reaction to the next reaction. And I'm like, okay, calcium, important. Good to know. These, obviously, because they have an entire positive or negative charge, are way worse than being polar, so they're not soluble in lipids. Again, ion channels are specific for the type of ions they transport. Now, when it comes to ion channels, some of these are always open, which means that the ions can just go in and out as the concentration changes, but some of them are gated. And next year, when I teach you about the nervous system and how nerve impulses are transmitted through neurons, you'll get to see exactly what I mean by gated. But in general, gated just means that they're closed until they need to open. And then there's some kind of signal that tells them to open.
Proteins form channels through the lipid bilayer. These channels allow certain particles to cross the cell membrane. Ions, such as this chloride ion, have a charge. Charged particles cannot pass through a lipid bilayer, but ions can move into and out of cells through a channel protein.
That is